Now we will discuss about the double helical structure of DNA. That means double helical structure of the nucleic acid we can take here. That means DNA is example of the nucleic acid. Hence we are DNA is showing the double helical structure. Hence we are taking the structure of the DNA as an example of the double helical structure of the nucleic acid particularly. If you take the DNA, the term always indicate what DNA it means deoxyribose nucleic acid first. DNA it means deoxyribose nucleic acid. DNA full form is always consisting of what? Deoxyribose nucleic acid, and RNA it means ribose nucleic acid. Both are the genetic material. Particularly DNA is consisting of a double helical structure. Double helical structure of the DNA has been proposed by the Watson and Crick. Watson and Crick both are the scientists. They have been proposed the double helical structure. Double helical structure of DNA. Here, double helical structure of DNA consisting of what? Two strands here. Double helical structure of DNA consisting of two strands. The two strands are always anti-parallel to each other. They are always anti-parallel to each other. Right? Anti-parallel. One, if the one strand is 5' to 3', other will be 3' to 5' here. And in these strands, particularly if you are thinking according to char graph rule, purines we had, pyrimidines we had. Just first we will see about the double helical structure, then you can get an idea. One strand we will take like this, and other strand we are taking like this. This is a double helical structure of DNA. In this thing, this strand, if you are taking this strand as a 5' to 3', and this strand will be 3' to 5'. And on this strand, what is happening here actually? The nitrogen bases are arranged here. How this nitrogen bases are arranged particularly? Like this. A is there, here the T will be there. Double bond. If T is there, then here A will be there. If G is there, here the C will be there. Here the C is there, here the G will be there. T, A, G, C. Like this. Right? Like this, the continuous in the strands, in the turns, this nitrogen bases are present here. Why we are taking A, G, T, all these things here? Because remember the point here actually, according to char graph rule, in the structure of the DNA, they are always consisting of a purines and pyrimidines. Purines and pyrimidines. Here purines are mostly two types here. Two types of the purines mainly consisting of what? Adenine, which is indicated by capital A. Guanine, which is always indicated by capital G. Adenine and guanine both are the purines. Cytosine is indicated by capital C. Then thymine is indicated by T. Then uracil is indicated by U. These three are the pyrimidines here. According to char graph rule, what is happening now? Purines will not bind. Pyrimidines will not bind here. Always purines binds with pyrimidines, and pyrimidines binds with the purines here. If you take the example here, adenine always binds with thymine, right? And guanine always binds with the cytosine here. It is a common phenomenon here, right? And in between the guanine and cytosine, we have three bonds here. Triple bond we have in between these two things. Double bond we have in between the adenine and then thymine here. Always the arrangement will be like this only, right? That's why we are arranging in the strands like this. A is always consisting of complementary T. T consisting of complementary, it means opposite. It means A. G it means C. When C here we are getting, G we are taking there. In the RNA, in the place of the thymine we are taking the uracil. In the RNA, not in the DNA particular. That's why we are not considering the uracil here. And particularly here what is happening? Turn we are saying. This complete thing we are calling as a turn. Double helical, two strands are there, right? Particularly the angle between this turn we are calling as what? 36 degrees will be the angle. Angle will be 36 degrees angle will be there. And if you see the base pair length between these two things, the base pair length will be 3.4 angstroms. Between the two base pairs, the length will be 3.4 Armstrongs here. If between the two base pairs the length is 3.4, in the complete turn the length between the base pairs will be 34 Armstrongs here. 34 Armstrongs. The length of the pair will be what? The length of the base pairs completely this turn will be 34 Armstrongs here, and 10 base pairs are present. For every turn, mainly 10 base pairs are present here, right? This is about the particularly double helical structure of the DNA, and A DNA we had, Z DNA we had, B DNA we had. Based on the DNA, again the base pair number is changing. Some will be consisting of 10, some will be consisting of 11 base pairs, some will be consisting of 12 base pairs. This thing we are calling as a base pair. Here also we can get the same thing. And very important point here, always structure of the DNA always consisting of a sugar phosphate backbone. It is giving support. Sugar phosphate backbone. Structure of DNA always consisting of what? Sugar phosphate backbone. Backside it is consisting of what? Which support the structure of the DNA, that is a sugar phosphate backbone. This is about the double helical structure of the DNA proposed by the Watson and Crick.